Dear students, how are you? Yes, we are going to a serious, dangerous situations. Yes, I'm talking about novel coronavirus. Don't worry. Stay safe at home and wash your hands regular proper time. Take your 20 seconds for washing your hands using 99.9 alcoholic hand sanitizer and keep social distancing. And we are using a common transport system, public transport system. Then we are reaching your house and your dress will clean in soap solution, and after cleaning your dress with soap solution you just entering the house. Okay, keep social distancing and avoid unwanted traveling to that such type of dangerous area, the prohibited area. We were avoid and we are aware about what is the disease, what is the reason for disease, what are the mode of spreading. So, and then we're back to our lesson. How do organisms reproduce? That is the lesson name. We are already learned the basic part of the lesson. And the organisms will reproduce because they're keeping their population, their number of the organisms increasing for generating the exact copies of that organisms. The reproduction methods, mainly two types: sexual reproduction and sexual reproduction. You know about two type of reproductions right there. In previous class, we are just starting the asexual reproduction methods. That asexual reproduction methods are including, first one is fission. Fission is already studied, but just recollecting fission. Fissions are two types are here. At the favorable condition, that cell first is within this cell the nucleus is divided into two, and then the two part of the nucleus are translocated towards the pole of that cell. Then cytoplasm will divide and form as two cells, two equal cells in favorable condition. Example, amoeba. This type of division is known as binary fission. Fission means cut into two equal parts. So, producing the exact copies, that is known as this type of binary fission. And then these two cells will dividing rapidly. The two is then dividing to form four cells, and 4 to 8, etc. That is the chain division reactions, like chain division reactions. And the second fission, the mode of reproduction fission, the second is multiple fission. Multiple fission means within the cell the nucleus divided into two or more than two nucleus, and then each nucleus is covered with a sheath with the cytoplasmic inclusions. During unfavorable condition, the cells within the cells, the nucleus will divide into more than two nucleus, and each nucleus is covered with sheath with the nuclear cellular cytoplasmic inclusions. And this nucleus with cytoplasm, that means small size of cells, these are dispersed at a favorable condition. Example, plasmodium. Plasmodium cause malaria. Plasmodium vivax, plasmodium malaria, plasmodium falciparum are main examples for plasmodium. Okay, then next is the fragmentation. Fragment means this compartment, this parts of one single organisms. That is fragment. Some of the alga means it's a chlorophyllated photosynthetic organisms, but these are not plants. These are under the category of plant kingdom. These are not under the category of plant kingdom. They have no leaf-like, no stem-like, no root-like structure. But these structure, these were formed like a thread-like structure. Within the thread, there are more chloroplasts are there. So, presence of this chloroplast helps photosynthesis. The photosynthetic fibers, photosynthetic threads are this in algae. Example, Spirogyra. Spirogyra is one of the examples for algae. They were seen in fragments, these thread-like structures, and they can reproduce with fragmentation methods because their body is cut into some fragments, and that fragments will grow with other organisms. We go to other organisms. The third one is the regeneration. Example, planaria. Planaria is the major examples for regeneration. Regeneration. The examples for regeneration is planaria. What is the structure of planaria? Yeah, about head and this is the structure of planaria. Then what is the regeneration? This planaria will cut into more than two parts. This is one part, this is two part, and look, one is this, and second one is this, third one is this, and fourth one is this. What is the regeneration? In planaria, this part, that is this part, will grow in a head part and base part and tail part. Therefore, another planaria. Okay, and then the tail part will grow, then appearing the next part and the other part and the third part. This is the regeneration. And next reproduction method is budding. This is planaria, this is regeneration. This is the budding. Main example is Hydra. Budding. That means in an organisms, they at growing stage, they produce some smallest growing part in their body. That is known as buds. Look, in Hydra, the body parts, the tail part and the dendritic part is the mouth. In Hydra, some of the external structures are involved. This is known as buds. This buds is separate from the main body, and will arise next organisms. So this is the another method of asexual reproduction. Then vegetative propagation. Next is vegetative propagation. Vegetative propagation. That means the vegetative part of the plant body. In a plant body, the body parts are commonly known as vegetative parts. The vegetative parts of the plant bodies are leaf, stem, root, etc. Leaf, stem, root, etc. So the other organism, other plant, that means the next generation is origin by vegetative propagations. That vegetative propagation means in leaf, the main example is bryophyllum. Have you heard of bryophyllum? Bryophyllum. In the leaf propagation, leaf originated to another plant, that is known as vegetative propagation with leaves, through leaves. And let's jump. The most of the plants are arising, the buds are arising from the stems and root. And some of the plants like sugarcane, what is the structure of sugarcane? What is the structure of main stem of the sugarcane? Look at that. This is stem of sugarcane. Some root-like structures are here. Root-like structures are here. From this part, are using the method of another agricultural technique, that is layering. For this stem has a root-like structure, but actually this is root. But this root will active in contact with soil. Then active as a root. And this type is a root-like structures. This is the another examples of vegetative propagation. Some banana, orange, roses, and jasmine are have the not able to creating next generation. That means that type of organisms, that type of plants, banana, rose, jasmine, and some oranges have the not their capacity to produce seeds. Such type organisms have been erased from budding, grafting, layering methods. These are the vegetative propagation. And Next is the important one. That is tissue culture. Tissue culture. What is tissue culture? What is tissue culture? Have you known about tissue culture? Yes, if you have first a notebook in proper biology notebook, take your notebook and give some examples for write some examples for this fragmentation, vegetative propagations, and some examples for regenerations, etc. Okay, you must write and learn well, study well. And next is the tissue culture. Tissue culture means grown by removing plant parts, removing tissue in placed in artificial medium. Tissue culture means grown by removing tissue. Grown by where? Removing tissue or separating cell. Tissue or cell, or by separating cells from growing part of the plant. From growing part of the plant. From where? Growing part of the plant. Look, tissue culture means the grown by removing tissue. Grown by, this is a process of grown by removing tissue, or by separating cells. Or by separating cells from where? From the growing part of the plant. From the growing part of the plant. Okay. Placed in an artificial medium, this tissue or cell from the growing part of the plant body. From the growing part of the plant body. Were placed into an artificial medium. Into artificial medium. Artificial medium. The tissue was dipped into artificial medium. Artificial growing medium, and then help to rapid division. And we help to rapid division. And helps to rapid division. That process is help to rapid division. That means divides rapidly. Okay, then this cell or tissue into artificial medium, then put into artificial medium, and will helps to dividing rapidly. They form a group of cells. Is called as callus. That means cell group. That is known as cell group. Then this cell group is transformed. This cell group is transformed. Then transformed. The cell group is transferred. The cells is transferred a medium contain hormones for growth. A medium hormone contain medium, hormone contained medium. That helps, these hormones will help growing. This is the artificial method for growing this. This is a cultivating method. Look, tissue culture may be defined as it is a process of cultivation of plants from growing part of the plant body. First is we collecting some tissues. We are collecting some tissues or cells from the growing part of the plant body. That is dip into artificial medium. That artificial medium is known as agar. It's a jelly-like structure, agar. And this agar have nutritive substance, nutritive substance for growing. The plant growing with presence of some nutrients, minerals, etc. These contain medium, that is agar. This hormone contained medium is also a agar, that's the next medium. Then, in this artificial medium, the cells or tissues will rapidly dividing rapidly. Rapid divisions are possible. And then see how we can see the cells of group. The group cells are here, that's known as callus. That callus is transferred to another artificial medium, that means hormone contained medium, that is agar medium, and then growing. And the growth of differentiation is possible at there. The growth of differentiation are there. Growth of differentiation. Growth of differentiation. And then this plant body is transferred to soil. Okay. These are very important because these are in disease, these are growing within the disease-free condition. These are growing in the condition of disease-free condition. Most of the ornamental plants are cultivated by this method because most of the tissue culture plants are in grown disease-free condition. Okay, this is the first. Then what is a tissue culture? Yes, you just write a question. That question is, what is tissue culture and why tissue culture is using the growing of ornamental plants? That's the question. Okay, we will discuss last class. And next, a sexual reproduction method is known spore formation. That is spore formation. Spore formation. Within the fungus, example rhizomes. The structure of rhizomes is you can see this rhizomes, this fungal is moist breadcrumbs. It's a grayish part. Some of the blackish in color. they can spreading fast because the division is very rapid divisions, very fast divisions are here. So this is the another essential reproduction method, that is spore formation. There's in rhizomes there is a sac-like structure at the tip of the plant. So this is not a plant, the plant-like structure at the tip of a sac-like structure. That is known as sporangia. Sporangia. The spores containing sac-like structure is known as sporangia. That sporangia dispersed and the spores were spreading through air, water, etc. This is very fastest asexual reproduction methods. Those are present in fungus only. The main examples of this spore formation is fungus. Then, now we can conclude in this type of reproduction. What is this reproduction? This is known as asexual reproduction. The definition is they can produce from single parent. Single parent produce younger ones. That means there are no difference between two male or female separately. There is not separation of male and female. They can originating from single cell or tissues. That means single parent progenies. That is asexual reproduction. Asexual reproduction methods are here, say the budding, fission, vegetative propagation, regeneration, spore formation, etc. This is the main points of the asexual reproduction methods. Yes, we just discussing all questions about this topic. Just write and answer it. The questions are: names the asexual reproduction methods. First question is name the asexual reproduction methods. Name the asexual reproduction methods. Yes, we already said that the asexual reproduction methods are fission. Fissions are two types, binary and multiplication. And the second one is regeneration. Third one is fragmentation, and the vegetative propagation. We are artificially next asexual reproduction method is tissue culture. That is in under the vegetative propagation. And the last is spore formation. These are the main examples for the methods of asexual reproduction methods. Next, the second is give an account of tissue culture. Give an account of tissue culture. Tissue culture means the artificially making the younger ones, artificially developing the younger ones using the method of vegetative propagation. First is we are collecting a tissue from the growing part of the plant body and then dip in a medium, that is agar, for the rapid division. And then after the division, they can see the group of cluster of cells are here. The tissues, the cluster of tissues or cluster of cells are here. That is known as callus. And this callus is transferred to another hormone contained, nutrition contained medium, that is agar. They can grow in that medium. They have nourished that other is very nourished with nutrition and minerals, etc. And this growing part of the plant body is transferred to soil. That process is called tissue culture. That method is known as tissue culture. And then within the tissue culture, what is the importance of tissue culture means? In after tissue culture, the developing from tissue culture, the plants were disease-free condition. Plants were in disease-free condition. And most of the ornamental plants are made with the tissue culture method. Okay. Then the next third question is, what is spore formation? Spore formations are within the fungus, the group of fungus. They can reproducing asexually by the method of spore dispersing. Examples: rhizopus. Rhizopus are seen in bird mold, the moist area, etc. They have a strong, long, slender-like thread-like structure, and the tip of the thread-like structure is a bulging part, that is a sac-like structure, that is known as sporangium. And in this sporangium, they can produce dispersing some spores. That spores will arising some fungus. So this is the very fastest spreading methods, that is fungal method. Okay, these are the asexual reproduction, because asexual reproduction is a developed origin from a single parent one. This is asexual reproduction. Okay, these are the questions, and you must write this question in your notebook and answer this question. And then thank you for cooperating with me, and bye. See you tomorrow. Bye.